एडमिन महोदय, सर ने ज्वाइन कर लिया है। सर
डन सर।
नमस्कार।
हेलो अंकुर जी,
सर
कैन यू
नमस्कार,
लिसन
मेरी
टू
आवाज
मी?
आ रही है? सर आप मुझे सुन पा रहे हैं? मेरी आवाज आ रही है?
एक मिनट एक मिनट एक मिनट, जरा मैं वो
ओके सर, ओके सर, ओके सर।
आपकी आवाज आ रही है, मैं जरा इसको बोस को जरा सेट कर रहा हूँ, एक मिनट।
हाँ हाँ, आराम से, बस मैं जानना चाह रहा था कि सुन पा रहे हैं आप सर?
बोस का मेरा ये सेट नहीं हो रहा है इसमें। अब आप सुन रहे हैं अंकुर जी? हेलो।
जी सर, जी सर, जी सर।
वेट ए मोमेंट, ये हेडसेट कनेक्ट करके देखता हूँ।
हाँ सर, आराम से
एक
सर।
मिनट, अभी आपकी आवाज नहीं ठीक से आ रही है। ये मेरा हेडसेट बोस का काम नहीं कर रहा है क्या?
एडमिन महोदय, आप सुन पा रहे हैं मुझे?
यस सर।
नहीं, आपकी आवाज स्पीकर से,
अच्छा।
लेकिन मैं ये सोच रहा था, ये मेरा अगर ये काम करे ना हमारा बोस का,
जी
तो ज्यादा अच्छा।
जी 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 जी, कर लीजिए सर, टेस्टिंग कर लीजिए, अगर हो जाए तो बहुत अच्छी बात है सर।
एक सेकंड एक सेकंड।
जी जी जी।
ये दिखा तो रहा है चेंज हो गया, आपको आवाज आई मेरी?
हाँ सुन पा रहा हूँ सर, मैं सुन पा रहा हूँ आपको।
आपकी क्यों नहीं आ रही है, सवाल ये है।
सर
वाई योर इज नॉट कमिंग। एक मिनट, मेरा ये वीडियो दिख रहा है आपको? मेरा फेस दिख रहा है आपको?
हाँ
दिख
यस
रहा
सर,
है?
सर।
देख पा रहा हूँ सर,
यस
देख
सर।
पा रहा हूँ,
यस सर।
हेडफोन्स को भी देख पा रहा हूँ सर।
आपकी आवाज लेकिन इसमें बोस में क्यों नहीं आ रही है, ये मैं सोच रहा हूँ। एक सेकंड, जरा मैं देखता हूँ। स्पीकर, एचपी, हेडफोन, ये हेडफोन है। आप बताइए कैसी आवाज?
क्लियर आवाज आ रही है सर।
राइट, आपकी भी आ गई मुझे आवाज, सॉरी
जी
डियर।
जी
ग्रेट।
जी, नमस्कार नमस्कार स्वीकार है सर।
चलो कोई नहीं, ये सब तो होता ही है भाई, आप लोग हाई टेक हैं, मैं नहीं हूँ। एनीवे, सो अंकुर जी, थैंक यू वेरी मच। सो शुड आई स्टार्ट, एंड हाउ मेनी आर देयर? हाउ मेनी पीपुल आर देयर?
चल रहा है सर, 100 के 100 क्रॉस करेगा धीरे धीरे, मैंने ग्रुप में मैसेज कर दिया है।
कोई बात नहीं, कोई बात नहीं, अगर कमी हो तो अच्छा है, कोई चुनाव नहीं लड़ना है मुझे। सो ओके, थैंक यू अंकुर जी, एंड organizers of this meeting, thank you
जी,
very much.
नंदिनी जी इंट्रोडक्टरी रिमार्क प्रोसीड करिए सर का।
अच्छा, ओके,
जी।
करिए।
Sir, am I visible and audible?
हाँ, यस यस, यू आर परफेक्टली ऑडिबल एंड विजिबल, प्लीज प्रोसीड इंट्रोडक्टरी रिमार्क।
Yes, thank you.
Okay, sir. Hello, everyone. A very warm and lovely good afternoon to one and all present here. I welcome you all on 30 Days International Training come Winter School on the topic Navigating Agriculture Transformation for the Upliftment of Rural Excellence, Nature 2024. I am Nandini Sharma, moderator for today's session. Today is 30th day of our training organized by AgriMeet Foundation Bharat in collaboration with as knowledge partner ICADA Lebanon, Southern Federal University of Russia, CIMMIT Mexico, World Agroforestry Center for International Forest Research Kenya, ICRI SET Hyderabad, Manage Hyderabad, SKL TSHU Telangana, MGUVV Durg, HAU Hisar, IGKV Raipur, IERI New Delhi, MHU Karnal, NDRI Karnal, ICR, IERI New Delhi, ICAR DCR Puttur Karnataka, Stanford University, and University of California. It is my absolute honor and privilege to welcome you all to today's session. We are gathered here to celebrate and learn from the exceptional contributions of a towering personality in the field of agriculture and genetics, Dr. A.K. Joshi, sir. Dr. Joshi is an eminent figure whose remarkable journey spans over 4 decades of groundbreaking research, impactful teaching, and transformative leadership. As a former professor at Banaras Hindu University, he has inspired countless minds and cultivated a legacy of excellence in genetics and plant breeding. His association with the CGIAR system for 16 years and his current role as CIMET Asia Regional Representative and Managing Director of Borlaug Institute for South Asia since 2016 have further solidified his reputation as a visionary leader. Dr. Joshi's monumental contributions include the development of nearly 100 wheat varieties in South Asia and Africa, addressing critical challenges like climate resilience, disease resistance, and biofortification. His commitment to advancing agriculture science is evident in his numerous accolades, including the prestigious Jeannie Borlaug Loeb WIT Mentor Award and the V.S. Mathur Memorial Award. Beyond his research, Dr. Joshi has been a guiding force for students, stakeholders, and farmers, sharing his knowledge through over 200 journal articles, extension manuals, books, and symposia proceedings. His work reflects a deep dedication to transforming agriculture into a tool for sustainability and global food security. Today, we have the incredible opportunity to hear from Dr. Joshi and gain insights into his pioneering efforts in wheat improvement, climate resilient agriculture, and genetic biofortification. Let us make the most of this session by engaging deeply and taking inspiration from his journey of service to science and humanity. Once again, welcome to this enriching session. Over to you. The session is yours, sir. Thank you so much.
Thank you, Nandini ji and Ankur ji. I think introduction was little heavier than my body weight. थोड़ा ज़्यादा हो गया, but anyway, not bad, not bad, not bad, you know. Okay, thank you. So let me share the PowerPoint. And Ankur ji, can you tell me the type of people who are attending this program?
BSc, MSc, PhD scholar, assistant professor, associate professor, professor, dean.
Hey, bhai sahab, so you have a very big range. Anyway, so this is a very big range. I was trying to understand this so that I can fit my talk, which I have kept quite general. So, dear friends, I have kept my talk a little general type with some examples what we have done together. And I selected this topic that fostering collaborations to resolve critical issues. In crops, certainly, because we are in agriculture, I'm in crop breeding. I have taken examples from wheat because I have worked on wheat, but there are countless examples in different crops. And this picture actually shows collaboration. You can see flags of different countries. These are the visitors who come to Mexico, the headquarters of CIMIT. So according to the number of countries represented, we show the flags. So if Indians participate, Indian flag is always there. And this collaboration is very important. Why I have selected this topic, dear friends? For simple reason that some people think that they can do many things alone. You cannot. It is almost impossible. So that's why I have taken this subject so that young people should understand the meaning of collaboration. Unless and until we have collaboration, we cannot benefit, we cannot show our potential, we cannot solve problem quickly. You can solve, but it will take much longer time. And if you see that artificial intelligence overview of the Google says that there are so many advantages. You can see a new perspective, efficiency, intellectual companionship, networking. You can have breakthrough research. You can do more ethically. So the scope is generally wider. So there are several advantages which you know, but when you really start collaborating, then you will feel the benefit of that. So this actually let me go to एक इसमें marker मुझे मिल जाता ना without a pointer. Pointer कहाँ से लाऊं इसमें भाई? Who can help me?
एडमिन महोदय आप बताइए।
If I can get the pointer here, थोड़ा इजी हो जाएगा, but चलो कोई बात नहीं, that's okay.
Sir. Okay.
हाँ बताइए, you are saying something?
Yes, I think from draw option it's over there, na sir. Like
ऑप्शन दिख नहीं रहा है इसमें. ऑप्शन के लिए मुझे बाहर निकलना पड़ेगा क्या? वेयर इज द ऑप्शन? वेयर इज ये इसमें होगा ऑप्शन. राइट राइट. वीडियो, पार्टिसिपेंट, चैट, शेयर, पॉज, साइट कंट्रोल, शो
no
मीटिंग
sir, in
मोर.
PowerPoint, there is option insert of draw option, na.
पावर पॉइंट, वेयर इज दैट? पावर पॉइंट में कहाँ है ऑप्शन? आई एम अनेबल टू सी इन पावर पॉइंट. मैं उसकी बात कर रहा हूँ, पॉइंटर की बात कर रहा हूँ. आई एम टॉकिंग ऑफ द पॉइंटर, नथिंग एल्स. वो उसमें रहता है. एनीवे चलो कोई बात नहीं है. नॉर्मली इट इज इन द जूम. एनीवे, so collaborations can be with different individuals or institutions or subjects having different purposes. You all know, sometimes we ask question, to whom we collaborate. Sometimes we think, oh, I will collaborate with my friend, I will collaborate with CIMIT, or I will collaborate with IRI, ICR, some other university, etc. It can be anything. It's not necessarily an institution. It can be subject. It can be collaboration with a farmer. It can be with a politician. Yes, with a politician, a policymaker. You can collaborate to understand the problem. Our body itself is a collaboration. So this picture you can see, our body is what? It's total collaboration. Millions of collaborations are inside our body. So dear friend, the collaboration can be of national or international for a problem, a big problem. You want to solve it together. Subject can be a new disease or something on nutrition or a crop management, engineering, or anything. It can be anything. And the beneficiary can be a farmer, it can be a scientist, it can be industry, even a policy maker can be beneficiary, or entire society, everybody. Now you may think how. Now there are examples. Sometimes new policy makers take decisions, green revolution. So policy makers were successful. So there are several examples. So I will tell you some success stories of collaboration in wheat, and I will see how fast I can go. I will not go very fast. I think we are on time. So I will give you this example. First is international collaboration for wheat blast. Now, do you know wheat blast? If you don't know wheat blast, see this picture. Collaboration is with India for Bangladesh, Zambia, Paraguay, Brazil, Bolivia, Argentina, because they all are suffering from a disease called wheat blast. Now, what is this? This is wheat blast. Now, wheat is looking very normal, beautiful. This picture is from Dinajpur, Bangladesh. I mean, the colleagues have given, although it is from Jessore. So wheat is looking perfectly normal, but actually there are spikes without grain. That's why it is called blast. Within 3 days, if weather is favorable, if pathogen is there, variety is susceptible, within 3 days, a beautiful crop will get converted into spikes without grain. This is wheat blast. Now, wheat blast was first discovered in Brazil in 1985. What does it mean? It means this disease was not present in wheat. Please mute yourself. Blast was present only in rice. For the first time, it emerged in wheat in Brazil in 1985. Now it is spread to about 3 million hectares. It can cause disease loss up to 10 to 100%, depending upon situation. So it was reported in Brazil, then it went to Santa Cruz region of Bolivia, then Paraguay, then Argentina, then observed in Bangladesh in 2016 and Zambia 2018. And because it came to Bangladesh, we are very much worried. India got worried because from Bangladesh, it can enter anytime. It doesn't require your passport. And if you see, what is this picture? This picture, if maybe you have seen this earlier, I got this from my colleague Pawan Singh in Mexico, but you can see it shows vulnerable areas in South Asia. If you see Bangladesh, from here, the mapping shows that it can move to other regions of India. And mostly it is showing central India, peninsular India. And central India is very important place for wheat blast. This is based on today's calculation based on last 5 years. It can move to north also. It can go to China. It can go to Japan. They are investing research in this. It can go to Nepal. So then we started a collaboration that for screening Indian wheat germplasm, we started using precision phenotyping platform for wheat blast in Bolivia and Bangladesh. Right hand is Bolivia. There are two centers, Okinawa and Curicilas. You can see that Japanese names. So Japanese went there. That's why the name is like this. And other one is in Jessore, Bangladesh, very close to our border. So we have created two platforms for testing. This is international collaboration. That India is collaborating with Bangladesh, also CIMIT, and through that we have a platform in Bangladesh and Bolivia. And this is the platform right-hand side, and you can see the impact of collaboration at Jessore, Bangladesh. Because of this, we have tested more than 10,000 entries tested and resistant variety released in several countries. Bangladesh, they have released varieties like Barigom 33, Vimri Gom 3. Actually, it is Bimri, BWMRI. Now, Nepal, Borlock 2020, Bolivia, Inifab, Tropical Iniaf, Okinawa. India, more than a dozen. There are several examples, DBW 187, HD 3249, etc., etc. So this is a very successful collaboration, international collaboration to control one disease. And this is the first variety which was released in Bangladesh, first blast resistant variety of wheat. It was released next year, within 1 year. Normally, we know that breeding takes 10 years. But why it was released within a year? Because already a top class line, which was good for all traits, we had no idea it has wheat blast resistance. We tested in Bolivia. So as soon as disease was noted in Bangladesh in 2016, we took thousands of lines to Bolivia and found that one variety which is already in advanced state of release was wheat blast resistant. Because this variety, Kachu solala, this variety is Kachu solala, and this is also biofortified. So it has high zinc. So this variety was under release for high zinc. But then by testing in Bolivia, we found that it is also wheat blast resistant. See the beauty of the collaboration. The beauty of collaboration is we were releasing for something else, and then we found it is good for another trait also, which is very important, wheat blast. So this variety was released. And then because of these studies, you can see this picture from Bolivia. You can see a picture, two wheat lines. One is dry, other is still green in maturity. So left one is completely susceptible. This is completely susceptible. And this is wheat blast, and right hand is susceptible, resistant. So this resistance we later found that because of a simple translocation from Agilops ventricosa. This is called 2NS. So it shows 85% less infection because of one segment, and it carries many other genes. And now we have molecular markers. So you can see collaboration happened at several levels, and finally we are successful. Now I will tell you another simple example about which you must be aware, some of you, but if you are not, this was actually like an education to policymakers. Now, how to collaborate with policymakers and why? For investment. Unless and until policymakers are aware that something is important, they will not invest. So education or collaboration with policymakers led to considerable investment and collaboration for UG99. You all know what is UG99. It is Uganda 99, which is actually a stem rust, you know, pathogen race. You can see left hand UG99 resistant, right hand UG99 susceptible. I remember taking this picture about 10 years ago in Kenya, and in right hand side you can see 100% yield loss is possible. So in farmers field, yield loss up to 80 to 100% was happening in Kenya, where wheat grows throughout the year. Now, right hand source picture I have taken from Science Journal, you can see the difference. If there is disease, it is completely dry. Other one is the green. So wheat blast, sorry, this UG99 was just like wheat blast, very serious disease. So UG99 is a race of the stem rust fungus that was significant threat to wheat production and food security because it could cause yield loss up to 100%. Now, this is the picture of this stem rust resistant gene resistant race, sorry, stem rust race. Now, if a variety is susceptible, it will look like this. Now, it has several disadvantages or threats. That vulnerability. UG99 was virulent, means it was able to attack a large number of genes and varieties. It was possible for this pathogen to spread by wind or in our shoes or clinging to our clothes. And that time, about 90% of wheat varieties grown worldwide were susceptible to UG99. So it was a very big threat. So it could have caused huge losses if it travels to other countries like India. That's why the world leaders got worried. And then this picture you can see, maybe you are present here. If you are present, you can locate yourself. This is a group picture of Borlaug Global Rust Initiative picture that was taken in Mexico. So through Borlaug's name, using the name of Borlaug, an initiative I started which was called Global Rust Initiative. So Borlaug started asking for money, and because of that money, the BGRI has bred hundreds of resistant wheat varieties that are now being grown at scale in priority wheat growing regions. So varieties were developed, they were spread to different countries, and now they are being grown. So, and because of that investment, see that investment is equally important. Sorry, I did that because the screen, yes. So because of that enhanced investment and collaboration, several resistance genes were discovered. So it was not only breeding, finding the genes that can be introgressed, that can work against UG99. Then a number of genes were discovered. Of course, some were discovered out of this funding also. I'm not saying everything from here, but this is an example that how through investment and collaboration, and not in one country, they have been discovered by different scientists in different countries under different conditions, different regions, different locations. So this is the beauty of collaboration. And then UG99 was blocked by resistant varieties. Before the disease could come, you can see how many varieties. This is not complete list. This is an example we developed 4 or 5 years ago, that in India, Bhutan, Bangladesh, Nepal, Pakistan, Afghanistan, Iran, Egypt, Sudan, Kenya, Ethiopia, in all countries, resistant varieties were grown, developed, and they were started to be cultivated. Therefore, when pathogen was supposed to come, it was believed that now it cannot spread. So this is a beautiful example how a disease can be blocked by resistant varieties. Because my dear friends, please note down, a resistant variety is like a vaccine. A resistant variety is like vaccine. And you saw the disease COVID-19. When COVID came, without vaccine, thousands and thousands of people died. So we wanted a vaccine to become resistant. So many people that time who were not believing that UG99 will spread, after COVID-19, they believe, yes, it is possible because they have seen a huge problem caused by COVID-19. Almost everyone got that infection. So you cannot stop pathogens. They have their own lifestyle. They have own evolutionary system. They are very powerful. The natural system is very powerful. Now, number 3, collaboration between medical and agricultural science. Now, this is a very beautiful example of collaboration between medical and agricultural science. That is zinc deficiency in human beings. Now, zinc is very, very important. Zinc is extremely important. This was not known earlier. This became known when Indo-Gangetic plains started showing zinc deficiency in 1990s. And breeding for biofortification was explored that time because the medical people said that we have to give pills of zinc. Now, pills of zinc, everybody cannot eat every day. So they said that, can we improve zinc in your crops? So zinc-rich varieties were released from 2016 onwards. So this work started around 2009-10. Actually, maybe before that, but it was not successful. This program started around 2005, 4-5, but it was not successful till 2010. Again, it was started in 2010 with more funding, and it became successful, you can see, within 6 years. And now, zinc is now a mainstream trait in wheat. Mainstream trait means now we treat zinc, in high zinc in grain, as a trait which should be present in 100% varieties. This is called mainstreaming. So, success story, you can see biofortified wheat and maize, and same thing happened in other crops also. So I am giving you one example which was developed by Mike Listman in 2019. So at present, about 34% wheat seed in India belongs to biofortified varieties. And it is the success of the combined efforts of medical, genetics, and policy. So medical and agriculture, that is genetics and breeding, also agronomy, and policy. Without policy, it's not possible. Our Prime Minister himself announced biofortified wheat varieties and rice varieties and other maize varieties he launched recently. So you can see the importance of being accepted by not only common people, also by policymakers. Now, fourth is collaboration with farmers. It led to understanding the need for early sown wheat. Why it happened? Farmers demanded early sown wheat to escape terminal stress without compromising yield. Why farmers were saying this? Point two, if you see, since pumping of water is generally banned in February in many parts of India, especially central India. If you are from Madhya Pradesh, you will see, if you read newspaper carefully, you will see that a notice in February, maybe second week, the district magistrate will issue a warning. Anybody seen pumping water will be under legal proceeding. So then it was an education to breeders and geneticists. Oh my God, this is the problem. We knew about terminal heat tolerance. We knew that yes, some varieties, short duration varieties are to be known. But if you see the last point, there were no knowledge about juvenile or early heat tolerance. There was no knowledge. Then what we did, then early heat tolerant lines were developed having juvenile heat tolerance. You can see the right hand field, early sown, late sown. This picture is taken from Obregon. It has a different purpose. Its purpose was terminal heat tolerance. That late sown, when it will mature in March, you can see one line is maturing, left hand. Other, when it will grow, temperature will already be 40 degrees centigrade. So how we did? We did by, you read point 2, by simply screening a large number of advanced lines under October sowing. Suitable performing lines were used for crossing with other best lines, and selection was done under early sown conditions. It's not a rocket science, very simple. We planted more than 3,000 lines, advanced lines in October. Success rate is always 5%, 2%. That's enough. So we identified these lines have early heat tolerance. No rocket science, no knowledge. Don't go deep into molecular immediately. Try to find out answer for farmers first. So we found the best varieties and they were released. And early heat tolerance is a new success story. By October sowing, it was proved that by simple change, plant 15 days early, there is around 1 ton yield gain in wheat. 10 quintal per hectare more extra yield you will get. Simply by planting early. And this is the example when we planted 540 lines under a normal zone in 3 years, you can see what is happening. The normal zone is dotted line. Below you can see the yield. Their yield potential is lower. And the darker line you can see yield potential is higher. So average you can see their average is about 7 ton. Here the average is about 8 ton. So these are the best lines from Simit planted in India. They were planted in India, in Ludhiana, Bisa, not any other place. And then 3 varieties were released in 2020. These are the 3, then half a dozen more since then. And now it's a routine. And this is the first wheat variety, DBW 303, which is first 8-ton variety. 8-ton average of 3 years means in some locations it gave 9 ton, in some locations it gave even 10 tons. So this is the beauty of collaboration with farmers and collaboration with partners to solve a big question in a very simple manner. So this is how we did that. And then early sowing became a policy intervention also in Eastern India. That early sowing of wheat gives enormous advantage. A policy change has been accepted by in Bihar, where the policy was that don't plant before 25 of November. They always used to say don't plant before 15 November. So 15 to 25 November used to be a normal zone condition. That we showed them that no, you can plant early and you can see what is happening. When you are planting in the first week of November and in the end of December, you can see the yield is higher there. So plant as early as possible, and this trial was conducted in 3,967 sites over past 5 years in collaboration with Kisi Vigyan Kendra in Bihar and Eastern UP. This is not example of 1 site or 10 site or 5 site or 20. 4,000 site, 5-year average. Data. This is the power of data. And that's why data is important. So dear friends, now the last I want to show, last but one. Do you know what is blue baby syndrome? कहीं देखा है blue baby? बिहार में देखा है, उड़ीसा में देखा है, कहाँ देखा है? ईस्टर्न यूपी में देखा है, साउथ में देखा है, नॉर्थ में देखा है? नहीं, in United States. It is because of water pollution caused by nitrogen. When babies drink water with high level of nitrate, it enters the blood. It prevents the blood from carrying oxygen properly, so causing the body to become oxygen deficient. So the white babies become blue babies. This problem was reported in many parts of United States of America. Also in Europe. Then it was found that fertilizers are the source of nitrate in the water bodies. If you want to live in Europe and do farming, you cannot do farming without preventing the leakage of your fertilizer in water bodies. They will take sample of water and then they will take you to the court. Police will come to you. Now they have legalized this system. So what from how nitrate goes to water? Because we apply ammonia. हम खेत में क्या डालते हैं? यूरिया. यूरिया क्या है? अमोनिया. It becomes nitrate because of nitrification. Because in soil there are millions of nitrifying bacteria. It's a microbial process that converts ammonia to nitrite and nitrate. You all know, agriculture students know about it. Chemistry people know about it. I am just trying to refresh you that there is a nitrification process all the time going inside the soil where you apply the fertilizer. This knowledge corresponded with awareness about genetic solution of this problem. Then people thought if we can stop nitrification, this problem can be stopped. But how you will stop? By neem coated urea or some other chemical coated urea. But then they thought, no, let us have a biological solution. BNI, biological nitrification inhibition. And they found this here. In 1983, research scientists found in Colombia and Brazil that fields of this grass is Brachyuria. It's a tropical grass also known as creeping signal grass. When they took samples here, they found there is no nitrate. They found that in these soils, there is no nitrate, very little nitrate. So they understood there is something in the grass which is stopping the process of nitrification. There is something. Then some colleagues found this grass, Japanese colleague, Lamus racemosus. 2 is equal to 4x is 20 odd genome. This is NNXX. It's a species of perennial wild rye. So wheat can be crossed with wild rye. It is also called mammoth wild rye. It carries the trait of BNI. It means the roots of this plant, they release some chemical which stops nitrification bacteria to work. ये biological solution है, ये antibiotic नहीं है. It is not antibiotic. It will release some chemical to slow down the activity of nitrification bacteria. Then a paper was published by Japanese colleagues and Dr. Subbarao. Dr. Subbarao is in Japan for 25 years. He published a paper, Can biological nitrification inhibition genes from this grass can combat nitrification in wheat farming? 2007. Now Subbarao, an Indian, he was selected by Japanese team by open competition. You can also be got selected in any country by open competition if you have innovation, if you read, if you think, if you discuss, if you collaborate, if you talk to others, and you develop a scientific temper. So Subbarao was not in Japan. He was outside Japan, not in India. He was in USA somewhere. And he was picked up in open competition. And then he did research and threw a beautiful question. And then we started this collaboration. How? Like this. So Lamus racemosus, the genes, a chromosome segment from this was transferred to wheat. Pre-breeding for BNI under CIMIT Japan collaboration, about 8 to 10 years. I'm writing 10 to 18, it can be 8 to 18 or 9 to 18. The pre-breeding was done and a chromosome segment from here was transferred to wheat. And then we found that BNI can work in wheat. Then we discussed in Mexico with Japan team. I discussed, actually, this is my collaboration. I discussed with Dr. Subbarao and Ravi P. Singh in a breakfast. We were taking breakfast, 7 a.m. in the morning. And then I said, I want to carry these lines to India and start breeding work with our Indian collaborators. He said, yes, why not? Ravi Singh said, Ravi Singh was that time head of breeding of Global Wheat Program. They said, yeah, why not? Why not? I immediately carried that material to India and then submitted a project in JICA, and it was awarded through Open Competition 2021 with a strong support of ICR. Even Niti Aayog supported this. And then we started doing shuttle breeding, discovery, molecular marker teamwork over last 3 years. There are 3 ICR centers. One is Indian Institute of Wheat and Barley Research, IIWBR. Other is in Karnal, same institute, CSSRI, Center for Soil Salinity. Third is IRI. And the pathway was like this. 2020, pre-bred BNI lines were imported from Civil Mexico, grown at BISA. Pure seed was obtained. And then 2021, BNI lines multiplied, shared with 3 ICR institutions. 2022, BNI lines evaluated at each of the 6 sites, ICR and 3 BISA. And 2024, a parallel breeding program was initiated by BISA and each of the 3 ICR institutions. Beautiful collaboration. A history of 25 years of research. Not a rocket science. Simple science, slightly technical, but you can do it. And now we know that BNI wheat can save 30% nitrogenous fertilizer because in the root they will stop ammonium to get converted to nitrite or to nitrate. And that's why you can see the pre-bred material, Borlaug, BNI, non-Borlaug. Non-BNI, right-hand side. The right-hand side doesn't have BNI, so it is looking yellow. It is looking green. Why? Because it's soil, it is preventing that bacteria to work. And now this is picture from CSSRI, Center for Soil Salinity Research Institute. You can see we tested these lines in 5 gradients, 0 nitrogen, 25, 50, 75, and 100% nitrogen. 3 BNI lines, 3 parent, 3 local check. And you can see the lines. You can see that if you read it carefully, below is nitrogen dose. You can see the BNI, these 2 lines were showing superior performance. BNI Rolf is dark green, BNI Vorebe is dark red. They are better than non-BNI in all fertilizer conditions. And these plots are very big. They are plot size 21 meter square. They are not 2-row, 3-row plot. Big plots. And same thing happened in BNI Munal after middle point. Well, nothing is perfect. It cannot happen in all lines because genetic background effect. But then it was proved. This is proof of concept. This is called proof of concept. Yes, it is working. And then the current state is that stages of breeding population now within 3 years. Some crosses are in F6, some are F5, some are F4, some are F3, depending upon back crosses. And now about 3,000 advanced lines are under evaluation. Other parameters are also being taken care. All traits. Best line will be screened for UG99, also for wheat blast. A BNI wheat may be in national trial in next 2 years. This is the beauty of collaboration. That suddenly, when people thought this is impossible, but it happened because of collaboration. So there are many other success stories of collaborations in wheat and other crops. Now name the crop, name the machine, name the country. There are collaboration. Whether you are partner or not, that is important. So you have to think, where are you? What are you doing? You are hiding everything. Oh, I don't want to share with anybody. No, you should discuss. This includes research publications. I will give one example. My own collaboration. Basic studies are very important. So we did some basic studies through collaboration, and I searched my publications between 7 to 20, only 13 years. I found that I have published more than 100 publications with national partners, where 50%, about 50%, 50 publications, 46% are on spot blotch, 9% on zinc. Then there are other subjects. So I'm telling you, spot blotch. Because I did collaboration with a pathologist. When I was young like you, maybe you are much more younger. But when I was young, I was struggling with my research. I was in genetics and plant breeding. I thought to collaborate with a pathologist, Dr. Ramesh Chand, not Niti Aayog, Ramesh Chand, who retired as a director of Institute of Agricultural Sciences, Banaras Hindu University. He was working in pulses. I said, no, you work, but can you collaborate with me for one Disease only? He said, what? I said, spot blotch. This is the disease. I want a pathologist to work with me because I don't know the pathogen. I know the wheat. I can do some crossing. I can understand genetics, but I don't know pathology. We started collaborating and then we solved major questions on pathogen side. The disease is a complex? No, it was not a complex. It was a myth. We proved it is not a complex. It was all publication was a myth. There are only one pathogen that was bipolary sorokiniana. Simple science we did. Then epidemic population and biology was not known. We said no, it is now known. We know the genes involved in secondary metabolism, virulence, were undefined genome. We know no, it is possible. Survival of pathogen was not known. People used to think, oh, where it will survive? It is in the air. No, seed is the main source. Then biochemical, histochemical traits were not linked to resistance. We said, no, no, no. They are linked. We can detect resistance. There are several publications. I am showing you only up to here 2018. But on host side, means wheat side, resistant sources, they say there are no resistant sources. I said, no, no, there are several sources. We published a series of publications. We have done molecular mapping. Genetics, they say unclear because there are several pathogens, so we don't know. We say, no, it's polygenic with 3, 4 genes are there. We have published a series of publications. There were no markers. Now there are markers available. We have done genomic selection. We have done so many things here. Molecular breeding, we have done molecular breeding. We have developed improved CV. So we have done all these researches only because of collaboration of two subjects, genetics, plant breeding, and pathology. Of course, there were earlier two scientists. On one side, I was, other side, Ramesh Chand. But then there was a team. Then several people emerged in from my team and his team, and we all were successful. So I want to conclude, my dear friends. This is my personal example. I want to tell you all that collaborations are necessary for doing high impact research. You should do collaboration. This is crucial for everyone involved, from scientists to policymakers. Don't think that you have not to talk to policymakers and you have not to talk to farmers. You have to talk to everyone. Even you have to talk to medical people, physics, chemistry, biology, social scientists. Go and talk to them. Somebody will tell some problem existing somewhere, and then your collaboration can lead to numerous success stories and groundbreaking proof of concept studies, and you will become a famous man. So my dear friend, this is also collaboration picture from Masai Mara, Kenya, where I visited once, and it's a beautiful place. I hope you will also visit one day there. Thank you very much. Yes, please.
Thank you so much, sir. I would like to take a moment to express my heartfelt gratitude for this insightful session. It was truly self explanatory and packed with valuable information. Even I have learned so much that will help me both personally and professionally. Thank you so much from our entire team of AgriMeet Foundation for sharing your knowledge and guiding us so effectively. Your efforts and expertise are deeply appreciated. Once again, thank you so much. Wishing everyone continued success
एक
here.
भी क्वेश्चन का सर डाउट नहीं रहने दिया आपने, एक भी
नहीं,
क्वेश्चन का, एक भी
अगर
लाइन का.
कोई क्वेश्चन है तो मैं सेवा में हाजिर हूं. अगर इफ एनीबडी वांट्स टू आस्क एनीथिंग.
जी जी, किसी पार्टिसिपेंट्स को यदि पूछना है तो आप पूछ
Yes.
सकते हैं. सर अभी 3 मिनट के लिए अवेलेबल हैं.
Yes, 3:57. Yes, I told I have to go somewhere at 4 o'clock. I have told them I might be late also. If
जी जी.
anybody has any question, Hari Kirat Singh Sidhu, sir, how can we just start breeding at our farm and undergraduate don't have any findings to? Are mere bhai. I think this is a very nice question. How can we just start breeding at our farm and undergraduate don't have any funding, so collaboration become difficult? It's great to listen. You are right. You are right. Collaboration is difficult. When I was young, very young, I joined as assistant professor in BHU. And there was a big meeting. Everybody was complaining, I have no facility. Sir, I don't have laboratory, I don't have field, I don't have laboratory, I don't have this. And everybody was complaining. And I was sitting quietly because I was very new. I thought I should. तो मैं चुपचाप बैठा हुआ था कि मैं बोलना मेरा अच्छा नहीं है, सीनियर लोग हैं। लेकिन उन्होंने देख लिया, हेड ऑफ डिपार्टमेंट ने। उन्होंने कहा भाई आप कोई शिकायत नहीं कर रहे हैं, आपको कुछ चाहिए क्या? मैंने कहा मुझे कुछ नहीं चाहिए। बोले क्यों? मैंने कहा सर लाइब्रेरी जाने के लिए कोई पैसा चाहिए क्या? दैट टाइम लाइब्रेरी वॉज वेरी इंपॉर्टेंट। आई एम टेलिंग यू 1985। मैं 1983 में साइंटिस्ट बन गया था, 1985 में। पुरानी बात बता रहा हूँ। मुझे 40, आई हैव एक्सपीरियंस ऑफ 41 ईयर्स। आपकी तो एज भी शायद 20-22 साल होगी, उतनी एज भी नहीं होगी। मैंने कहा मुझे कुछ नहीं चाहिए। मेरे मुंह से निकल गया कि मुझे लाइब्रेरी जाने के लिए भी कोई पैसा चाहिए क्या? बड़े लोग नाराज हुए मुझसे। बताइए, हैं? कल के लड़के पता नहीं क्या क्या बोलते हैं। माय डियर फ्रेंड, इफ यू डोंट हैव एनीबडी टू कोलैबोरेट, स्टार्ट कोलैबोरेटिंग विद लाइब्रेरी। हाफ ऑफ द रिसर्च इज डन इन द लाइब्रेरी। वेयर इज लाइब्रेरी? अरे पूरा वर्ल्ड मोबाइल के अंदर है भाई। रीड वन बुक, वन पेपर, एक पेपर रोज पढ़ो। एक्चुअली एवरेज 3 होना चाहिए। नॉट यू कैन फिनिश 90 पेपर इन 1 month। You read 3 paper. Next day again you read it. Again you read. When you travel, take a book. आजकल फ्री में बुक मिलती है। कहीं जाइए, दिल्ली में चले जाइए, चांदनी चौक में 50 रुपए की, 20 रुपए की किताबें पड़ी रहती हैं, ले जाओ। प्लीज डेवलप हैबिट ऑफ रीडिंग। हम लोग पढ़ते नहीं। ये एक, आप पढ़ते होंगे। आई एम वेरी ग्लैड। ये क्वेश्चन मुझे बहुत अच्छा लगा एक्चुअली। It's a very nice question. Somebody is actually asking question. It means, Hari Kirat ji, you have a desire to do something. कोलेबोरेशन आप अपने आप से स्टार्ट करो। आपके अंदर भी एक आदमी बैठा है, उससे आप देखना आपको नॉलेज बढ़ेगी। व्हेन यू विल स्पीक, 10 आदमी आपसे इंप्रेस होंगे। दे विल कम टू यू, दे विल स्टार्ट कोलेबोरेटिंग विद यू। आई थिंक दिस इज द वे वी हैव टू स्टार्ट व्हेन नोबडी इज देयर। यू आर करेक्ट, बट स्टार्ट रीडिंग एंड देन टॉकिंग टू पीपल। और आप कर सकते हो। और किसी के पास अगर कोई प्राइवेट, अगर कोई स्टार्टअप शुरू करना चाहता है ब्रीडिंग का, सपोज आपके पास लैंड है एंड यू हैव सम मनी, यू फील दैट आपके फादर ठीक ठाक हैं या आपके दोस्त हो, आप कर सकते हो। कोई बिजनेस दुकान लगा सकते, चल रहा है। अगर ब्रीडिंग करना है, easiest way of doing breeding is to take material from a organization like CIMIT। आप हमारे विसा फार्म में आइए, हमको बताइए, हमें अच्छी लाइन दीजिए। आप ब्रीडिंग क्या करना है? आपको टेस्टिंग ही तो करना है। फिर उसकी जो अच्छी हो, चेक से आप मार्केटिंग शुरू कर सकते हैं। थोड़ा रूल्स रेगुलेशंस का फॉलो करना पड़ेगा। किसी भी सीजी सेंटर से आप मटेरियल फ्री में ले सकते हैं, सब आपको मिलेगा। आपको सिर्फ डेटा देना हो। सो देयर आर वेज ऑफ डूइंग। स्टार्ट में मुश्किल होती है, मैं मानता हूं, बट इवन आफ्टर कैरियर ऑफ 10 to 15 ईयर वी डोंट वांट टू कोलैबोरेट। तो प्रॉब्लम होती है। छिपाना नहीं कुछ। जो नॉलेज है दूसरे को दोगे तो वो बोलेगा नहीं 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 नहीं, यू आर रॉन्ग हियर। आपको पता नहीं है, बड़े बड़े महान गायक जब गाते थे ना, तो एक बच्चा आके कहता था आप गलत गा रहे हैं। लता मंगेशकर ने अपने पिताजी को कहा था आप ये ने ठीक से नहीं गाए। तब उनको लगा ये बच्चा इतनी बड़ी गायिका हो सकती है। सो दिस इज हाउ वी हैव टू कोलैबोरेट। थैंक यू।
बहुत बहुत धन्यवाद आदरणीय। अब तो मुझे ऐसा लग रहा है कि कोई क्वेश्चन नहीं है। हर व्यक्ति का क्वेश्चन का आंसर जो है एक लाइन में मिल गया है कि उन्हें लाइब्रेरी जाना चाहिए। वहाँ हर एक चीज का त्वरित व्यवहारात्मक निस्तारण है, उनको सबको लाभ मिलेगा उससे। तो बहुत बहुत धन्यवाद सर। आपके पास भी समय का शॉर्टेज है। मेरे जी
एक पॉइंट और,
जी
मेरा ईमेल
जी
दे दीजिए। इफ
जी
एनीबडी वांट्स टू कोलैबोरेट विथ मी, कनेक्ट
हम्म,
विथ मी,
जी
प्लीज फील फ्री टू राइट ईमेल। आई विल बी एट योर सर्विस ऑल द टाइम। जो भी मुझे आता है मैं आपको बताऊंगा। थैंक यू।
जी 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 आदरणीय, मैं जरूर ईमेल लिख दूंगा जो ग्रुप है हमारा पार्टिसिपेंट्स का उसमें। सर बहुत बहुत धन्यवाद और बहुत बहुत आभार। मतलब धन्यवाद शब्द ही नहीं है सर ज्ञापन करने के लिए कि आपने मेरी रिक्वेस्ट को एक्नॉलेज किया और लेक्चर को सर डिलीवर किया इतनी व्यस्तता में भी सर। बहुत
ठीक है,
बहुत धन्यवाद।
प्लेजर। थैंक यू, प्लेजर। थैंक यू।
जी
बाय बाय।
जी बाय।
बाय बाय। थैंक यू।
मीटिंग एंड कर दीजिए रोहित जी।
Okay, sir.